will say about functional assembling and self-replicating peptides. So why this assembly? Why this process of aggregation? Why is forming an out of entropy process? So before going into the main topic, I would say something about the order out of Q. It's a big term. We cannot say it in a one line. We cannot describe it in one sentence. It's a process in which a kid makes toys from Lego bricks, the unorganized substance, and after organization, it comes into life. In nature, you can see geological patterns where the minerals, the sediments, they are aggregated in a very nice pattern to produce something which we see in our daily life. In floral pattern, where the leaves are arranged in such a way, an order structure is formed. In the galaxy, in nebula, a huge amount of particles, with huge amount of energy, they form nice patterns, they form nice structures. So it's assembly. So it's the process of aggregation, and it's the process of forming order, it's forming a pattern. In human culture also, there is production of pattern, and pattern which we can see the historical artifact, we can see the modern art where the pattern forming is an integral part of art. So they are at the same time functional in nature. Like if you see the color of a morpho butterfly wing, and if you go in detail, like in microscopic imaging, you can see there are patterns in the wing which scattered the light, which scattered the blue light to make it blue in color. The assembled structure of tobacco mosaic virus, which is an aggregation of RNA and protein molecule to give a functional entity, a living entity. Like cytoskeleton, where protein aggregate to form tubule, and this tubulin protein forms integrity in the cell, and therefore we can see rigid structure, rigid cell in our biological system. So, which molecules form aggregate? Generally, they are called amphiphilic molecules. So, the name amphiphile implies, obviously, those who are from undergraduate and postgraduate in this lecture, obviously you know about amphiphilic molecules. The molecules have two parts. They have philicity towards both phases, aqueous phase as well as hydrophobic phases. So their polar head group that attracts water molecules, and their hydrophobic tail triggers the aggregation. They form the hydrophobic aggregation process. Consider a soap molecule. Soap molecule have hydrophobic head and the hydrophilic head and the hydrophobic tail. Hydrophilic head get dissolved into water and the hydrophobic tail get aggregated with each other to form this kind of core. And this kind of core can encapsulate oil. This core can encapsulate the dirt from your shirt or your t-shirt, and it can get chunk out from the cloth and can come into aqueous phase, and then you wash it out. Glycocholic acid, it's a part of membrane proteins and cholesterol unit that is present in the living cell. It's hydrophilic and hydrophobic at the same time. It stays in membrane, gives it stability and do its biological work. Phospholipid membrane constructing lipids. They are also important because they have polar head group, they have apolar tail groups. The head groups attracts water and the tail group repels water. So this love and hate relationship make a bilayer very much stable, so we can get a rigid cell. And these molecules, when we study different type of aggregating amphiphile, we can find various structures. They can form bilayer, they can form micelles, rather reverse micelles here, vesicles, tubular micelles, nanotubes, fibrils, folded protein structures, viruses as I have shown you earlier, self-assembled monolayer and many others. So here two processes crosses each other, they work in an opposite function. Repulsion between the charge groups that is present in the amphiphile and the repulsion between the charge group and also from the electron in the molecule, and also an attraction from the different non-covalent forces. So what are the non-covalent forces? The non-covalent forces are electrostatic interaction sometimes, hydrophobic interaction, van der Waals forces, London dispersion forces, and hydrogen bonding interaction above all, which dictates the stability of this assembled structure. So why we have chosen peptide? If you see the structure of a peptide, they have this kind of plural amide bonds. So amide, then a group, then another amide, and overall a structure is formed, which is called a peptide structure. And here, if you can see, they have both hydrogen bond accepting and hydrogen bond donating groups. And this makes it very interesting. And in nature, we also see peptide with myriads of structures like beta sheet, town structure, coil, and many others. And it's a huge subject of protein folding, which deals with the non-covalent interaction formed by peptides. So I will present my work here on peptide-based hydrogel for the release of cancer drugs. So you know about cancer drug release because Obijita last time introduced you regarding the release of drugs and its interaction with the living cells. And we try to module the stiffness of this gel by incorporating B amino acid residues into the peptide structure. So before going into the main topic, I will say briefly regarding something about the drug release. So as Obijita told you, that the release is important. The slow release is important rather than the burst release. Like if you are doing a burst release, at a time you are getting a certain amount of efficiency, and then it diminishes with time, and you don't get enough efficiency after a particular amount of time. So it dies out. But if the release is controlled, then you, for a long time, you will get a sustained release. Slowly the drug will work, but steadily. And if you compare the sustained release with a pulsatile release, you will see that the area under curve of sustained release is obviously much more higher than a number of pulsatile release. And secondly, cancer drugs have side effects. As you know, doxorubicin, it also kills the regular cells. So we need to reduce the side effect. We need to prolong the drug action time, and we at the same time we need to improve the efficiency of the release. So the common delivering agent which has been reported are micelles, liposomes, nanoparticle, and carbon-based materials. Actually, these micelles or the liposomes encapsulate drugs into their hydrophobic part depending on the hydrophobicity or hydrophilicity of the drug. And the micelle in their hydrophobic coat, they encapsulate drug delivered into the targeted cell. Nanoparticles also do the same job when the drugs are attached with the surface as well as the carbon-based material. But many of them has various side effects. Like if you are using a micelle which is not very much biocompatible, or which is not very much, which interaction is not known very much, then you will see that a certain amount of toxicity is there associated with the micelle. And the carbon-based nanomaterial, if not delivered in a proper way, they can be toxic for the human cell. Therefore, we tried to find out a gel, a hydrogel matrix, from where an easy release of doxorubicin is possible. And at the same time, it has injectable properties. Like you can take it by a needle, you can do injection, and after injection, again it will form a gel inside your body. So, the interesting fact associated with this it should have the proteolytic stability. If it is proteolytically unstable, then after a certain amount of time, you will see that your gel has lost, your component has lost, and you have nothing in your hand. So you have to have a good gel, you have to have good proteolytic stability, and it should be non-cytotoxic at the same time. So I was discussing about gel. Why gel is so important. And what are the gels? So gels are soft viscoelastic materials. They can encapsulate the solvent, the matrix, which we generally use for making this gel, to give it a rigid shape. And how they make it? They form this kind of assembled network. You can see in the picture. And these assemblies can hold solvent molecule like a fishing net holds a fish. And by this way, it gives a shape to the overall structure of the solvent, and we get this kind of solid gel. Depending upon the structure of the gelator, we can classify it into two types, supramolecular gel and polymeric gel. So supramolecular gels are such that where small molecules are used for making these gels. And polymeric gels are polymer, normal known polymer system which are hydrophilic and hydrophobically balanced, can encapsulate the solvent. So in our study, we are using a small peptide molecule. So obviously we are using supramolecular gel, where self-assembly prevails in the system for making the gel. And depending upon the media, it's hydrogel, organogel, aerogel, or ionogel. So these are the categorization of the gel. So why they are useful? Because gel can be used in many fields like 3D cell culture, drug delivery, for using the radiological test, adhesive, optoelectronic material, contact lens, and removal of waste. And out of this, I today will discuss mostly on drug delivery, optoelectronic material, and removal of waste. So let's concentrate to the first topic. So we had this hydrogel, we had this peptide in our hand, which forms a tight hydrogel at pH 7.46, which is also the physiological pH. But it's not proteolytically stable because it has all L-phenylalanine residue, and the chymotrypsin in the body can easily chunk it out to make the gelator a non-functioning unit. So how can we tune? How can we keep the enzyme present in our body from cleaving, and how we can retain the gelation, how we can tailor the gelation property? So we can do a few things with this molecule. If you carefully see the molecule, there are CH2 units which are connecting the phenyl ring to the backbone. So phenyl ring, which is a good pi stacking which forms a good pi stacking interaction, can be changed. The interaction amount of interaction can be changed if we truncate this CH2 unit. So we can replace the phenylalanine with the phenylglycine. We can do reduction. We can do hydrogenation at this part. So this is no longer aromatic ring, it's a cyclohexyl ring. We can do in this way, or we can change, which is I think most easy way, change the chirality of the component amino acid. Replace the phenylalanine, L-phenylalanine with D-phenylalanine. So out of these three, we try to do with the last part, last trick. And for that, we synthesize all possible stereoisomers of the peptide, all L, BLL, LDL, and LLD, and their corresponding enantiomers. So what we saw, the all L at the same time, all D, it forms good gel, also DLL and LDL. But when we change the chirality of the end amino acid, then it cannot form a gel. So there is something obviously which is changing. So what we hypothesize that if we change the chirality of the system, maybe we can play with something, we can tune something. So this is really working because we are not getting gel out of one small change in the C terminus. And interestingly, they all form this kind of fibrillar morphology. So fibrillar morphology, if you go to the field emission scanning electron microscopy, it's a microscopic technique where you can see the intricate details of aggregation, intricate details of organelles under microscope. It's not normal microscope. It uses electron instead of light for using for looking at the molecular aggregate and molecular structures. So what we found out of this, we found that the release from the LLL was much lower than DLL. So the DLL is efficient in terms of drug release. On the other hand, similarly, because they are diastereomers, they are enantiomers, DDD shows similar trend like LLL, and LDD, which it shows the similar trend like DLL. So out of the 6, the LLL, DLL, and LDL and their enantiomer, we got these two and their enantiomer as efficient releasers, efficient drug releasing platforms. But out of these 4, this LLL and DLL, they are not proteolytically stable. They break down with time when we use a proteinase enzyme. We used here protease K during the drug release. We found that they undergo degradation and do not work. But DDD and LDD, they are proteolytically stable. So out of these 4, we have now these 2 in our hand. They are good platforms for drug release. But out of them, LDD is the most effective because it releases for a longer time, like 79 hours. So we are getting a good sustainability. We are getting a good release profile. So out of the all stereoisomer, after screening, we found this molecule. We concluded this molecule as the most efficient gelator system, and it's also injectable at the same time. Like I told you earlier, it should be injectable. You can take it out with a needle, you can inject it back, and you can form the gel. So it's thixotropic. I mean, it's forming, it's regaining its shape after you are injecting. We tried to do the study. We tried to find out why they are forming this kind of aggregated structure. So from the FTIR study, we found that the efficiency of formation of the beta sheet structure of hydrogen bonding, for LLL it is the max. For DLL it's lower, LDL is much lower, and LLD there is very weak hydrogen bonding interaction. So this again goes with the trend of our hydrogelation, the process of forming gel. And from the circular dichroism, we found that this is a process, this is a overall chirality of molecule. We see the chirality of the aggregates. And we found that the terminus, the N terminus, the C terminus, I mean the end point, dictates the chirality of the assembly. And here again I want to quote Brislov, who said that it's the chirality do not reside in the stereogen, it's the chirality of the whole molecule which matters. And here also the chirality of the whole aggregate which matters in case of circular dichroism. It's a very interesting spectroscopic study. It's a spectroscopic technique. You can read it out. We did powder X-ray diffraction. In the diffraction study, we generally see the distance, the distance between the sheet-like structures, and we found that for LLL, like the all L isomers, there was a good agreement with the aggregation study. The distance between the peptides were perfect, and for the other species, we did not find any peak for 3.8 angstrom, which we generally assign for the pi-pi interaction of the phenylalanine moieties. So this is maximum for the LLL. So what we tried to suggest is that obviously a change in chirality will change in the aggregation pattern of the phenyl ring. Yes, this is true. And this is the frequency sweep rheological experiment. This is a simple experiment. I am not going into the details of the physics. It's a big physical experiment because you are dealing with, when you are dealing with hydrogel, you are dealing with viscoelastic materials. So they are viscous and elastic at the same time. And you are basically comparing the viscous coefficient and the elastic coefficient by using this kind of study. The conclusion from this study is like, for say it's the rigidity of the gel, it's maximum. For first mutation, it's lower. For second mutation, it's much lower. And for the third mutation, you can't even see its rigidity because it's now solution. You can't say it's a gel. So the mechanical stiffness is also changing with the mutation of the point amino acid residue in our gelator system. And this is the Thixotropic experiment. What we say about the injectability, I am not going into detail. And yes, we found that this lead product, from the lead product what we got, the end product, LDD, it's non-cytotoxic in nature. It doesn't show any kind of cytotoxicity. And it shows a much better killing of cancer cell when we loaded dogs with the gel instead of free dogs. So in conclusion, we found a gel which can release something, which can release an anti-cancer drug, and we can use this anti-cancer drug for treating in a more efficient way. And at the same time, we found a way in which we can change our assembly structure. We can tune our assembly structure in a most promising way to make a most efficient releasing platform. So, do they only release drugs? Do they only release substances from their matrix? No. Gels are very important things. They have porous structure, so they can absorb things, they can release things. Yes, they can absorb things, like dyes from the wastewater. If you see the scenario of pollution, there are two kinds of pollution here I will discuss about. First is the industrial pollution from the organic dyes. So industries release huge amount of sludge, huge amount of organic dyes into water body, and from a nice illustration from WWF, they showed that the whole earth is drowning into the garbage tank because we are basically accumulating garbage every day. And second, another important process is oil spilling. So spilling of oil on the seabed, it make a huge impact on the marine life. The marine life get in danger when we spill oil like anything over the seabed. And reported a case of huge oil spill. So people are really worried about that. And a historic event, probably you all know that the example had a case where a huge amount of sludge, huge amount of oil spilled over the seabed, and in the coastal area of Canada still is facing problem due to this kind of oil spilling. So out of this, in which way we can change, in which way we can use our gel systems for this kind of pollution removal. So we can do one thing. We can use the porous system. We can use the porous matrix of a gel to remove the oil from the seabed, and at the same time we can use them to recover the oil spill from the system. The porous structure, they can hold the dye, they system. The removal of oil spill, it can be done by organogel, what I have discussed already. There you can put your gelator molecule, your gelator systems over the seabed, and it can collect the oil that was flushed over the bed. So in this regard, we found a gelator. This is a gelator molecule. If you go to the structure, it has polar head groups. These head groups can interact with water, and these parts which are hydrophobic in nature, they can form hydrophobic interaction and pi stacking interaction. At the same time, if you see the reverse case, if you take the solvent apolar like normal hexane, then you can see that they will interact in a better way with the non-polar solvent, and then here they will not try to get exposed to the solvent. And this process nicely was found in case of this gelator, as this can gelate a huge amount of solvent, starting from water at pH 7.5 to normal hexane, which is an apolar solvent. So this is the beauty of aggregation. This is the beauty of self-assembly and gelation, where you can use a gelator, single gelator molecule to gelate a polar solvent and a polar solvent at the same time. And this type of gelation, this is called ambidextrous gelator. So ambidextrous, dextrous means hand. So it has two hands. It can at the same time gelate polar and same time gelate apolar solvent from a system. And yes, when we studied the same image, scanning electron microscopic image of the gelator systems of the gels, we found that in all the medium they are forming network structure. And probably for due to more kind of interaction, more kind of ordering in normal hexane system, which is completely apolar solvent, there it forms a nice fibrillar aggregate. And from the FTIR analysis, we found that the extent of hydrogen bonding is much more better in case of a non-polar solvent like N-hexane. So basically what is happening when you are taking a molecule and you are trying to form a hydrogen bond, the media in which you are dissolving it shields the charge of the system and you are getting less amount of hydrogen bonding. Water, as a good dielectric media with high dielectric constant, it shields the charge, it shields the hydrogen bonding ability of the peptides, and at the same time it competes in hydrogen bonding interaction. When you are using an apolar solvent, where the head groups, which I have shown here, they are not interacting with the solvent, rather they are trying to aggregating with each other through the process of hydrogen bonding. So this is happening here. When we did a small angle X-ray scattering, we found that there is a nice intercalation of the peptides. So the peptides form a hydrogen bonded structure with an average length of 39 angstrom, which is less than double of the unit molecule. So they are forming basically pi stacking interaction and hydrogen bonding interaction at the same time. Moreover, the hydrogen bonding is so precise, you can see the 4.72 angstrom peak due to the distance in the two strands. And the small angle X-ray scattering nicely. Again, we did rheological property to confirm the process of gelation. I am not going into the details. And going to the, if you see here, like in case of acid black, we have loaded the gel with the solution of acid black. And after some time, we saw that the dye got trapped inside the gel matrix. So the gel matrix with its porous structure is basically doing the just opposite thing that we saw in our last case. It's basically absorbing things from the water in a useful way because it's removing acid black, a toxic organic dye from water. And now your water is clear with a very low amount of dye in the solution. So how did we measure it? We measured it using UV visible spectroscopy. So you can see a good band at around 600 nanometers for the dye molecule, and when it gets removed from the solution, its concentration is decreasing with time. Similar thing we observed for Congo red, for malachite green, as well as for rhodamine B. So these all are toxic dyes. These are 5 toxic dyes that frequently comes from industrial wastage, and it gets absorbed by this gel matrix. Again, you can recycle this gel. How you can do that? You can take the gel out, you can dissolve it in ethyl acetate or extract in ethyl acetate, you remove the ethyl acetate under vacuum, and then you get your gelator intact again. You dissolve it again and do your same experiment, same kind of treatment on water. So this is recyclable at the same time. And the important part of this is from the surface of salt water. So for this experiment, we did one thing. We took salt water, we put petroleum or palm oil, petrol, diesel loaded over the salt water. When we put the gelator molecule dissolved in ethyl acetate into them, we found that they form a gel. I would like to show you a movie in this which we made for this experiment. So if you see, yes, this is forming gel. So these are two phases, and I am adding gelator in ethyl acetate. And you see, immediately it got blurred. I'm sorry, the video is not of good quality. And when you tilt it, yes, it has captured the oil. It has gelled the spilled oil from the seabed. So in conclusion, it can be said that you can use a gelator which is ambidextrous in nature, or both used for removal of industrial waste and removal of spilled oil from the seabed. And this spilled oil, how you can recover it? You can take this out, this layer out, you can evaporate it under a rotovapor to get the spilled oil back. Now I'll go to another topic. So we were so far discussing about the removal or absorption from the gel bed. Now we are going into the material property of this gelator system. So peptides, the molecules are conducting also at the same time. Yes, you know, in the bioelectronic system, in the system of biology, there are a number of proteins that can conduct electricity. They form spores, they difference between outer and inner phase of a cell, and they can do their job nicely using their residues, which can conduct charges from one point to another. Here in this paper, they show nicely how the charge gets transported from one domain to another domain of a Ramachandran plot through the process of charge transport of the residue. And the process is not very simple. It's either long range or short range in nature, depending on the nature of study. Like if you are doing it in electrochemical process, you are taking your protein or your peptide in a solution, then obviously the charge gets transported through the process of tunneling or flickering resonance process. And when you are taking them in the solid support, you are covering them between the two electrodes. You will see that there is a super exchange, the process of charge transportation, and the process of hopping. So basically, you are getting a conducting peptide. We are trying to getting a conducting peptide, which, like the biological system, can conduct the electricity from one point to the other. And you can read this article in this regard. It's a very nice review describing how the process of charge transport happens inside a peptide fibril in Journal of Physical Chemistry B. And here comes the process of organic supramolecular semiconductors. So they are a new type of materials where instead of molecular orbitals, due to aggregation process, they form valence band and conduction band. And if you feed the valence band with electron, it can easily get excited to the conduction band, and then it can drain the electron to the corresponding end of the electrode. So we are trying to, so the scientists are trying to get good optoelectronic material using this principle, and in this regard they found a very good candidate. This is called naphthalene diamide. This is a common class, this is a general class of Rilene dye where there is an aromatic core which forms a good pi stacking interaction. And this stacked molecule, they can transport charge, they can transport electrons throughout their aggregated structure to give us a good electronic device. In this end, we tried to make hybrid out of two systems. We took our supramolecular fibrillar system, we took TiO2 nanoparticles, which are also good conducting material, to make a photoconducting system with good efficiency. So you all know about the famous photoelectric experiment where when you irradiate a surface with light, you get an electric response in your circuit. So this is the same what we are using. We are trying to mix these two to get an optoelectronic response in a photo switching manner. So for this, we take this Rilene dye. This is called Perylene bicimide. This is a red dye, and we functionalize its end with a dipeptide, one from the unnatural amino acid and other from the peptide and non-amino acid like phenylalanine. So they form a nice aggregating structure because they have pi interaction site here and here. They have hydrogen bonding interaction site. They have hydrophilic ends. So it is expected that they will form a bola type of structure inside water. And yes, we found that they form good hydrogen. They form tight hydrogen when we try to dissolve this compound in pH 7.5. So why this is important for us? Because after initially was molecular band got into an aggregate band, and it is expected to now show a good conducting, semiconducting or photoconducting behavior. So we will see what is happening here. Yes, they have formed fibrils. These thick ribbons are obtained from the zero-cell of our peptide. And in fibrillar structure, TiO2 nanoparticle as shown in this black. TiO2 nano on this process, they are good photocurrent conducting things, and people have done many experiments on this TiO2 nanoparticle, and currently they are also used in solar cells. So we tried to infuse TiO2 nanoparticle into the gel matrix, and we found that yes, the nanoparticles are forming interaction with the thick fiber present in the matrix, because you can see that particles are decorated over the fibril. And what expected was the overall current would increase with the formation of hybrid. And yes, this really happened. If you see, it's a simple current voltage experiment where you change the voltage and you see what is happening with the current. And in this case, we made a device using a tablet. This is a tablet of either peptide or the nanoparticle or the hybrid system. In this, among these three cases, we found that for the hybrid system, the amount of current value is the highest, and it is very important to see that they had so low current response, and after hybridization, their nature changed entirely. So obviously, some synergy is in between them. Therefore, they are giving this kind of result. And the aerosol itself shows good photo-switching property. I mean, this is only PBI gel, no nanoparticle, nothing. So only PBI gel shows good current response upon irradiation of light. So you are switching light, you are getting a response, you are getting an increase in the current at a constant voltage, and when you switch off the light, the current decreases with time. Again you switch it on, it's increasing, and then it's decreasing when you are off. So this is called photo switching experiment. You are getting current response when you irradiate a device with light. So this is a gated process. And also in case of TiO2, you see there is a faint response, but there is a response. But when we mix them together, there is something so high. Now in the hybrid system, you are getting a huge amount of photocurrent response when you are irradiating them with the light. So why this is happening? Our explanation was that when you irradiate the PBI with the light, it gets excited to its excited state, and the excited electron or exciton can get transported to the conduction band of the TiO2 to make it more electronically active, more electroconducting. This is happening in our case, therefore we are getting so nice experimental result. And this is the sum up result. If you see the gain current, the ion by I off ratio is much higher for this kind of hybridized system. So yes, we are able to make a hybrid system which has some in between property of TiO2 and PBI, because only TiO2 shows this kind of wavelength response of current, and this PBI shows like this, and the hybrid system shows something in between and is showing a good amount of photocurrent property. So using self-assembly, using the process of molecular pattern formation, using the process of aggregation, we have found a device-like thing which we can use in future for making new systems. Now I am going to my current research, what I am doing here in Ben-Gurion University. So this is something different. Now we'll discuss something. I will not go about much details of my own research work, but I will say you some glimpse of why it is important, why we are doing these studies. So if you see the timeline of life, if you see the process of Darwinian evolution, you will see that there are three processes that prevail in a system of the process of life formation. First is compartmentalization. You need to squeeze your system in a confined environment so you can get the optimized result. So if you consider life as a chemistry, a process of chemistry, a process of out of equilibrium, and a process of order out of chaos, then you have to have compartmentalization. You have to have aggregated structure, what we have discussed in earlier slides. At the same time, another two important characteristics of life is there. First is self-replication and second is metabolism. Self-replication is a process where the molecule, the life replicates its own structure, its own entity. From a particular species, you are getting the same species with the evolving time. So this is the process of self-replication, what we say reproduction. And metabolism, it's nurturing its own quality, it's nurturing its own life through the process of some chemical swelling. And if you see in a cell membrane, if you see in a cytoplasm, you will see that compartmentalization is always happening there in order to promote the process of replication and metabolism. And if you consider the abiogenesis, one of the philosophy, one of the theory of life, how it evolved, you can see that from abiogenesis, from non-living organic small molecule, information giving systems like RNA and DNA got evolved, and slowly they got into a system, a confined system where they can replicate themselves and they can nurture their life through the process of metabolism. Then it came into human culture. I must also say about this, when the cognitive evolution of human began, and in culture they also tried to mimic the process of in nature, knowingly or unknowingly, through the process of digitization. They tried to store the information like RNA or DNA stored their genetic information in computer. In living cell they do in RNA, DNA, in computer. And yes, using self-replication process, they bring out their data and replication. And the molecule that participate in metabolism is protein and peptides, and the process in which the replication occurs is mainly the nucleic acid. So in prebiotic condition, consider when there was no distinction between amino acid, no distinction between nucleic acid. So what happened then? Maybe then it was not as hard and fast that DNA are the only replicating molecule. Maybe at that time peptide also participated in the replication. This is the famous work from Muhammad Reza Ghadiri, where he discussed about a process where an alpha helix can replicate its own formation through the process of ligation, native ligation. And for that, what he found that when men and put some random nucleophilic and electrophilic substance having similar sequence with this one, they found that after some time they formed good assembled structure, and this assembled structure gave rise to another helix. So he started with one helix, he got two helix at the end from his component. So this is the beauty of the self-replication of peptide. Maybe this was happened when there was no nucleic acid in the prebiotic soup. When there was only peptide, when only amino acid pool, nature collected all the peptide and amino acid into a long backbone of helix or beta sheet, and maybe it replicated its own formation. At the same time, this fibril can be conducting, because if you attach, if you very into this peptide, you can see they can be conducting. So what I told you earlier, conducting peptide nanofibril, and what I am telling you now, them both. We can combine them in such a way so that after replication they can form conducting bands. Like if you are taking this peptide as electrophile and this as nucleophile, maybe after formation of this ligated product, the array of naphthalene diamide present in the system can conduct electricity in the process. I am not going into the details. I am not going into the FRED process with which we are trying to monitor the replication. Yes, from tryptophan and NDI, there is a fluorescence resonance energy transfer, another fluorescent phenomenon. I am not going into detail. It's a matter of biological system where they frequently use FRED to monitor the chemical changes inside molecule. So we are trying to get a conducting fibril through the process of self-replication to get a good device where the replication can trigger an enhancement in the current generation in the peptide fibril. So at the end, I want to say thanks to you to give me an opportunity for giving this lecture. Thank you.
Thank you, sir. Thank you for your brief and lucid presentation. So, if there are any questions, I would like to from audience, if any questions you have.
Sir, I have a question.
You may ask.
Please.
Sir, what is proteolytically stable means?
Yes, proteolytic stability means when you are using a peptide, you are using a peptide for therapeutic application, no matter for what you are using. So proteolysis is a process where the protease enzyme that is inside your body chunks out this peptide. Protease enzyme is there in blood. It tries to digest the protein or peptide you are using in your blood. So it can take it out, it can chunk the protein, the peptide bond like this CONH bond, it can cleave. So you are starting with the tripeptide, after some time you will finish with end with a dipeptide, which has no activity. So this is the process of proteolysis.
Sir, I have another question. Sir,
Yes.
is there any role of gels in sewage culture? I mean,
In what?
sir, is there any role of gels in sewage culture?
Sewage, sorry, I can't. Sewage?
Yes, sir. Sewage culture. Sewage culture. I mean, a role of gels in sewage culture. I mean, is there any role of gels in sewage culture, sir?
No, it's not like that. Here we have only told about the removal of oil spill. You are telling about that?
Yes, sir.
Yes, it can be used for removing oil spilled over the seabed.
Okay, sir.
Can any further questions from the audience?
I have a curiosity. Can I ask that? Just
Yes, sure.
a layman's question: whether or not there is any method to directly assess or measure or quantitate self-assembly process or self-replication process in your experimental setup? Of indirect method. Just I was thinking of any direct assay method exists or not.
Yes, so when we are considering about self-assembly, so we are going to single molecule to molecular ensemble. Yes, you can do it using thermodynamic experiment, but I must say there is still now no direct method.
No direct instrument-based method, no, not possible. Right?
You can
Measuring
measure, you can monitor the self-assembly using NMR, using the thermodynamic technique like dynamics. So you can measure the kinetics of the assembly. Yes, you can do that.
Energy. Okay.
Yes, that in that way, kind of you can measure it, but still you need to using like fluorescence change. Like if you are using a fluorescent aggregating molecule, using a change in the fluorescence, like when aggregate it changes its fluorescent property, you can measure it. But you can say only about the kinetics of assembly, but you cannot say that this is happening right in this way. No, you cannot say it still now.
Thank you.
So can I ask one question?
Yes, sure.
So regarding this oil spill, after this hydrogel is formed, so this is, but if you want to really do it on a grand scale, like if a oil spill has happened and we put the gel into the water bodies, and how will you collect it and dispose it off from the water body? Because it is in small scale, it is viable.
It is viable. Yes, I completely agree with you. See, when you are using it a large scale, it is supposed that it is we did not do it obviously in large scale. We did it in small scale. You have huge amount of chunk of gel inside your on your water body. Then you can take it out theoretically and you can remove the water. You can remove the oil, but still we are under progress. Still we cannot say using this gel we can do everything. And still now the absorbent, the hay dust and many other things, they are the best way to remove oil from the seabed. Yes, we are developing, but it's not a complete, it's not a blueprint where you can, it's not in state of art. Yes.
So the cost effectiveness is also there.
Yes, the cost effectiveness also is there. Yes, you need to have a good production ability to make it large.
And suppose in your assessor you have carried out on different kind of oils, palm oil, that, but generally whatever this means this transportation, creosote oil or heavy oils are there, like C16 or C18 chain. So was it not better for any creosote oil kind of thing, because it is most transportable?
No, we did not actually see. Yes, yes. Actually, we did the experiment. If you see in the slide, we did it with diesel. We did it with petrol, and at that time we did not access to all the creosote oil what you are telling, or palm or other oil. We did not have access. But palm oil, I don't know its composition, but probably it is also heavy oil, isn't it?
It is C18, most probably.
It is CA.
Most probably, I'm not
Okay,
sure, but
okay.
could
So we
be.
did it with this only.
Okay, thank you. If anyone has any questions, thank you, sir. If anyone has any questions, kindly ask.
Thank you.
I believe there are no questions. So to conclude the program, we would like our monastic faculty member of Department of Industrial and Applied Chemistry, Ishatma Chaitanya Maharaj, to kindly give the vote of thanks. Maharaj, please.
Am I audible, Saurabh?
Yes, yes, Maharaj.
Okay. Om Namo Bhagavate Sri Ramakrishnaya. So, on behalf of Vidyamandir family and Industrial and Applied Chemistry Department, I would like to humbly thank our Principal Maharaj for his welcome address. Also, thank you Professor Pratap Kumar Das, former scientist ISCB, to be present with us and deliver the keynote address. I like to take the opportunity to offer our sincere thanks to our honorable speakers, Dr. Obhijit Saha, former postdoctoral research fellow, Institute of Chemistry, Hebrew University, Jerusalem, Israel, and Dr. Kingshuk Basu, postdoctoral research fellow, Department of Chemistry, Ben-Gurion University of Negev, Beer-Sheva, Israel. We are privileged to hear the enlightening and informative lectures from the speaker on very demanding topic of the modern application-based biochemical science, namely synthesis and use of peptide-based molecules for cancer treatment, targeted delivery of anticancer agents, and functional self-assembling and self-replicating peptides, which are obviously fully embraced with knowledge. I would like to thank our departmental head, Dr. Uttam Kumar Ghoroi sir, and all the faculty members, research scholars, and students of our department and Vidyamandir for arranging and attending such a beautiful program. Thank you.
Thank you. Thank you.
Thank you, Dr. Basu, Dr. Sinha. Thank you.